I will jump into our first presentation, and that is a primer, a genetic primer for the modern nephrologist. So it's a primer, a refresher for our nephrology colleagues and how they can use testing, diagnostic, and management in their patient population. So our first speaker and a dear friend of mine, Dr. Bianca Russell. So Dr. Bianca Russell is an assistant clinical professor in the Department of Pediatrics and the Division of Genetics at UCLA. She obtained her medical degree from UC Irvine and completed her combined pediatrics and human genetics residency and metabolic fellowship at Cincinnati Children's Hospital. Dr. Russell is a clinical geneticist with a particular interest in kidney conditions, including Fabry disease, polycystic kidney disease, kidney malformations, and other genetic kidney disorders. Dr. Bianca Russell, when she came to UCLA Health a few years ago, has changed, at least from my perspective, how we are managing genetic causes of kidney disease. She's also accompanied by her genetic counselor, Miss Marina Clarke, another very, very amazing counselor, but also a key member of the UCLA Nephro-Genetics program. So a bit of history about Miss Clarke. She's a genetic counselor, board certified by the American Board of Genetic Counseling and licensed in California. She received her master's degree in genetic counseling at University of California, Irvine, and received her bachelor's degree in biological sciences from Rutgers, the State University of New Jersey. Her role at UCLA is focusing on chromosome 22 mutations, hearing loss, kidney genetics, and general pediatrics. She is a member of the UCLA Clinical Genomics Center, where she assists in variant interpretation for exome and whole genome sequencing tests in both clinical and research settings. Marina is also a clinical supervisor and lecturer for the UCLA Genetic Counseling Master's Program. So with that, it's really my privilege and honor to welcome Dr. Russell and Ms. Clarke for their portion on primers. Thank you.
Thank you for the introduction. So I'm Marina Dutra-Clarke. I'm a genetic counselor at UCLA. And how we're going to set up this talk is I will start by giving you an overview of genetics, and then I'll hand it off to Dr. Bianca Russell, who's a clinical geneticist, and she'll share some really interesting cases, and you'll see how we've applied a lot of these genetics principles. And our talk is called The Genetics Primer for the Modern Nephrologist. And this talk was supported by Natera. So we know that genetics plays a role in nearly all aspects of medicine, but a particularly strong role in kidney diseases. And Dr. Rastogi just highlighted the many benefits of knowing the underlying genetic cause of the patient's kidney diseases. And so it's very important. I really want to emphasize that today. We've actually been in this genetic revolution or genomic era for quite some time now, though. We've sequenced the human genome 20 years ago, and next generation sequencing was developed 10 years ago now. So I think by now we really should be offering genetic testing routinely for these patients. And you've probably noticed how fast-paced the genetics field is. There are tens of thousands of genetic tests on the market, and with more options, it can be more confusing in selecting the right test for your patient. You've probably noticed that the list of genes on panels can vary between commercial labs. The test methodologies can vary between labs. So if possible, collaborate with clinical geneticists and genetic counselors to help select the appropriate test for your patient and for the right price for your patient. So after the sample's at the lab, how do they analyze the sequence data? Well, the lab is looking at your patient's variants, which are the sequence changes in the genetic code, and they're taking those variants and comparing them to the variants in our large data sets of other people in the general population data sets. So, and looking to see what is different in your patient. The problem with this is that there are millions of variants that differ between one individual and another. So human variation is normal. So it's very difficult to decide whether a variant is benign or a disease-causing variant. Most labs use this 5-point scale that indicates the likelihood that the variant is disease-causing. It ranges from benign to likely benign to variant of uncertain significance, or VUS, likely pathogenic, and pathogenic. And then I listed out some of the common tools and criteria that's used in variant interpretation and classification. So for example, population databases are key. Like if the gene is typically associated with a severe kidney disease and it's highly penetrant, then we would not expect that variant to be commonly seen in our population database. Also, is it the right variant for that gene? So for example, some kidney diseases are exclusively caused by a particular variant in a particular domain of that gene. So is it the right mechanism? And then has it been reported in the literature before? Has that variant been seen before? Is there co-segregation data? So often we'll order genetic testing for other family members and see, is that variant tracking with the disease in that family? And also, does the clinical and family history fit with your patient? Sometimes phenotype is not part of the variant level interpretation. So definitely use your clinical judgment and always send in your clinic notes to the lab. And what about the in silico prediction tools? What do they say? Those are the computer algorithms that can help predict the effect on protein function. And are there any in vitro or in vivo experiments for this particular variant? Because that would be very helpful too. And this is also why genetic test reports are 10 pages long sometimes, because they lay this all out for you, all the databases that they used and all the tools that they used. Hopefully in the future, our patients will have better access to orthogonal methods like RNA sequencing and proteomics, but for right now, you're pretty much working with this list. And something to really keep in mind is that some labs don't agree on a variance classification. So there is discordance. Even though they're all using similar guidelines, like standardized guidelines, there's still discordance. So here's an example. There was a study that came out last year where they took 8 labs, submitted variants and their classification, and nearly half of those variants had discordance between 2 or more labs. And 63% of the pathogenic and likely pathogenic variants were discordant with variant of uncertain significance by another lab. And 11% of the discordance cases would have impacted clinical recommendations. So here's a sample genetic test report that I want to walk you through. So starting from left to right, the gene is listed, a PKD1 gene. And the disease that it's associated with is polycystic kidney disease. And the inheritance is autosomal dominant. So we have 2 copies of all of our genes. And autosomal dominant inheritance is where there's a pathogenic variant in one of those gene copies. And then here in the variant column, that lists out the GPS coordinate of your variant. So here it's always given in 2 nomenclatures. So the top line is the cDNA nomenclature, and the bottom is what's happening on the protein level. So the cDNA, that's showing you which variant is changed. In this case, the variant was deleted. And then there was an effect on the protein. So we have the leucine is the first protein that's affected by this. And it's changed to a tryptophan. And in fact, this deletion of that one base pair caused a frame shift in the entire code. And it affected all of those amino acids, 93 amino acids further down, and then led to a premature stop. So FS stands for frame shift, and then the asterisk tells you that there was a stop. So this is a truncated protein product. And it's heterozygous, meaning they found that variant in about half of that sample, so consistent with that autosomal dominant inheritance. And the lab classified this variant as pathogenic, and they list out the rationale under the interpretation section. And I just outlined some key points in the interpretation section showing that it is the right type of variant, because as a frame shift, truncating variant, that does fit with the mechanism for PKD1. And also it's been reported in the literature in unrelated individuals. So that's showing replication. And then lastly, the variant is absent from our Broad Nomad dataset. So this variant is rare and not seen in our healthy general population. So now I've just explained this to you. How do you explain this to the patient without using technical terms and technical jargon? So I would just, I would keep it simple. I would just say, our genes are the instructions for the body to function properly. And one of those genes is the PKD1 gene. And there was a typo found in that instruction. So the 11,554th letter was deleted in those instructions. And that caused the instructions to stop short. And this is called autosomal dominant polycystic kidney disease. So in addition to just giving the patient the genetic information, what else would be important and part of the genetic testing, genetic counseling process? Well, genetic counseling is the process of helping people understand and adapt to the medical, psychological, and familial implications of genetic contributions to disease. And genetic counselors really like to emphasize the part that says and adapt. So it's not just giving them the genetic information. We have to also help our patients adapt to this information. So work through your patient's emotions. Address shame, guilt, fear, and misconceptions. There are some patients who don't realize that the age of onset or disease severity can vary even between members of the same family who have the same variant. Also, empower your patient. How can we increase your patient's perceived control? We can't change the fact that they have this genetic condition, but there are ways that we can help them with how they perceive the uncertainty of their risks, how they react to just having this genetic condition, and how they perceive the value of the medical treatment and interventions that are recommended thereafter. We provide medical and support resources. And of course, genetics is a family affair, so we discuss familial implications, like testing at-risk relatives, and we provide a family letter that they can use to distribute. And also for any family members who wish to be potential donors, we recommend genetic testing in them. And we counsel on the recurrence risk based on the inheritance pattern of the gene, and family planning options are discussed. For example, there is an option to not pass on the genetic condition to the offspring using in vitro fertilization and pre-implantation genetic testing. So with that, I'm going to pass it on to Dr. Bianca Russell. Thank you.
Great, thank you so much, Marina. So we're going to jump into some cases that are going to highlight some genetic principles. So we'll start off with polycystic kidney disease. This is a case of a 9-year-old male with left multicystic dysplastic kidney that was noted prenatally, and he has a solitary right renal cyst that was found at age 4. He is a di-di non-identical twin, and it was conceived by IVF with a sperm donor. So his genetic testing identified a mutation in PKD1, and this is the mutation that was identified. Interestingly, it is a truncating mutation, like Marina just described to you, a frame shift variant. But something a little bit different about this is that it's only present in 20 to 25% of the cells that were tested on this blood sample. So we call this mosaic, meaning that it doesn't affect every cell in the blood that was tested, but only about 1/4 of it that showed this specific change. So we see this in about 1% of patients with ADPKD, and we think that as well there's about 10% of ADPKD that does not have an identifiable genetic cause but fits clinically. We think about 10% of those cases may in fact be mosaicism that's missed. And that's challenging because we do not look at every cell in the body. So when we take a sample of the blood, we're getting only a small piece of what's actually going on in the rest of the body. And it may be possible that there are changes in the genetic code located just in specific tissue types, such as the kidneys. So in this young boy's situation, it's quite possible that there is a higher mutation burden in one kidney over the other, hence the variability that you see with one kidney having prenatal onset and the other having more of a classic ADPKD presentation. When we see a mosaic pattern like this, it is expected to be de novo, and that this is a change that was caused after conception. So a couple notes about PKD1 as a gene. It's quite challenging. It's very large with 46 exons. It has a high GC content, which means that it's really difficult to sequence. There are 6 pseudogenes, that are about 98% the same as the gene you're actually supposed to be looking at, and it's on the same chromosome. So as you can imagine, it's really challenging to sort out what is the pseudogene and what is the actual gene of interest. So it's really important to have full gene coverage, and you really do need specific sequencing tools and skilled interpretation in order to analyze variants in PKD1. And it is important to check whether or not the lab that you are using actually tests for this gene, as many labs do not include this gene on their gene panel due to the technical difficulty of interpreting and sequencing PKD1. Moving on to Fabry disease. So this is a case of a 34-year-old female. She reported GI dysfunction, fatigue, peripheral neuropathy, severe anxiety, depression, and progressive memory loss. There were no identifiable renal or cardiac conditions. And so here's just a picture of some of the systems that get impacted by Fabry disease, which is a lysosomal storage disorder with multi-organ system involvement. Her genetic testing identified a change in GLA, which is the codes for an enzyme that's important for breaking down waste products in the lysosomes. And this is the variant here that was reported in her. And if you look in the description of this variant, it's a little bit tricky, and there's a question of what this variant actually means. And they actually raised the question of whether or not this variant would be impacting clinical function based on how it affects the enzyme level and whether or not this could be a pseudodeficiency. So one of the tricks we have up our sleeve is to look at some functional studies, or in this case, metabolic studies, and look at how something like an enzyme is working in the body. So for this patient, we tested her enzyme level and found that, in fact, her enzyme level was low. But then we take it the next step to look at what would be building up as waste products in the cell or the substrate to see if there's a buildup, as you would expect with a storage disorder. And in this case, there is not. And so the number we look at is the lyso-GL3, and her number there is normal. So just as a picture of showing what's happening here, the blue cell there on the left is your typical unaffected individual. You're in the lysosome there, and there's this GL3 product that is broken down by an enzyme and then utilized within the cell. In somebody who has Fabry, you see that there's that big red X and the enzyme is not working as well as it should be. So you see buildup of lyso-GL3 levels. This then raises the question of what is this variant doing, if anything, right? So looking at it in more detail, it is a common variant in the general population. In vitro studies in publications early in the 2000s show that enzyme levels can be low on the enzyme assays, but it seems to be due to the substrate that's used to actually do the test, but not really resulting in true low enzyme levels in the body. But then if you look in the literature more recently, you see publications about cohorts of patients that seem to have some more neurologic-based features. And there's this question raised about whether or not there are, in fact, features of people who have this variant. But there are some challenges there as well, in that these are patients that were ascertained through cohorts of individuals who presented with neurologic features, particularly peripheral neuropathy and chronic pain. So it's tricky, right? It's hard to understand how do you interpret this and what's the next step. One opportunity is to test a tissue of the organ system that you're most concerned about. So doing something like a biopsy of the kidney and looking for storage within the kidney tissue can really help you understand in a variant whether or not there's disease that needs clinical management. There are medications, both oral and IV enzyme therapy, available for Fabry disease. So it's important to make sure that you have the right treatment or are not treating a patient that doesn't need treatment. Moving on to another kidney condition, we've got Alport syndrome. So this is the case of a 33-year-old female with microscopic hematuria that started at age 12. She has a strong paternal family history of clinically diagnosed Alport syndrome, and she herself has no vision or hearing loss. And we'll go through her family history next. But just to highlight what's going on in Alport syndrome from a genetic standpoint, there are 3 genes that are primarily associated with Alport syndrome. The X-linked formed, but as well as both recessive and dominant inheritance patterns. And I bring this up because it gets really tricky. You've got genes that the same gene can cause both a recessive and a dominant inheritance pattern. And it can be quite tricky to understand how your patient and that family may be affected by particular variants. So looking at this family tree, you see our proband, or the patient here, is the 33-year-old with the arrow who has hematuria. She has a brother with hematuria as well. Her father passed away at age 51. He was blind in his teens, had hearing loss that developed in his 30s, end-stage renal disease in his mid-30s, and renal transplant before age 40. He has 4 siblings, 3 who are deceased at younger than age 50, and all presented with similar findings as himself. Interestingly, his parents were both asymptomatic throughout their life. And in fact, his mother, who is 100 years old and doing well, has 4 children with another partner who are unaffected. So the genetic testing here did identify a variant in COL4A3. And again, this is one of those tricky genes where it's hard to know whether or not there's a dominant or recessive inheritance pattern for this variant, as both patterns have been reported. So Alport inheritance is one of the more challenging ones to understand. There can be reduced penetrance and variable expressivity, meaning that not every individual in the family presents the same, even if they have the same variant. Individuals with heterozygous COL4A3 variants can have benign hematuria and be considered carriers for autosomal recessive Alport syndrome. And patients' relatives, paternal relatives in this case, had an earlier onset disease more consistent with an autosomal recessive pattern, whereas our patient fit maybe more as a carrier or as more of the dominant form. It is possible that there may be more severely affected family members having a second variant in the COL4A3 gene, or it's been postulated that there can be a second variant in another gene, such as COL4A4, resulting in something called digenic inheritance. This is where 2 different genes that really work closely together can each have a change, and those things can combine and have a confounding effect. In this family, unfortunately, we'll probably never know because the relatives who were severely affected are deceased. But it gives us a good example and framework for some of the challenges with Alport. Next, we'll talk about focal segmental glomerulosclerosis. So this is a 55-year-old female with hypertension, type 2 diabetes, rheumatoid arthritis. She had severe renal stones with a history of obstruction and infection. She has diverticulitis, status post-colonic resection, and focal segmental glomerulosclerosis with associated nephrotic syndrome that's been quite tricky to treat. She has 4 healthy children in their 20s and 30s and 3 grandchildren, no family history of renal disease. So just as a background on FSGS, it's part of a genetically very heterogeneous group of nephrotic syndromes. The age of presentation is really highly variable, and there are over 40 associated genes. Large gene panel testing identifies a genetic cause in around 20 to 30% of the patients that we do test. So this is the results from her testing, and I'll point you, I guess, to first the 2 other genes listed below the gene in red, and these are both changes in recessive conditions without a second variant. So as Marina articulated and showed you the dominant pattern, this would be a recessive pattern where you would need a change on both copies of your chromosomes in order to have the condition, and she has only 1 of each of those, so we're not concerned about that. But this first one here is certainly of interest. So ANLN stands for actin binding protein anilin. This is a gene that's important in cellular growth and migration and helps with cytokinesis. It causes autosomal dominant focal segmental glomerulosclerosis 8 in just 2 families that have been reported in the literature, but it also has shown a phenotype in zebrafish as well as mice. Because only 2 families have ever been identified or at least reported in the literature, there's only 2 variants that we're aware of. And this makes it really difficult to interpret the variant, right? So it has not been previously reported. It's not common in controlled databases. It's predicted to be damaging in multiple protein prediction software programs and is the type of variant that may cause disease. So while the lab can't go to calling this a pathogenic variant and calls it a variant of uncertain significance, looking at the actual data, it's pretty convincing that this is in fact the explanation for this family. Next steps then go to how do you investigate this in the family? So we went on to test affected and unaffected family members. Her 76-year-old mother was negative for this variant and does not have renal disease. Her father was deceased, so we were unable to test him. All of her children were screened for proteinuria. Her 32-year-old son was found to have proteinuria and was positive for this variant on follow-up testing. And he is now undergoing surveillance and close monitoring. So the next step in this case is to collaborate with researchers and the folks who published those 2 families. We've collected patient samples and provided the variant information to those labs where they can look at zebrafish and mouse models and look to see how this particular variant in this family impacts other organisms. So I wanted to then finish up by talking a little bit about some family planning. This is a really important part of what we do in genetics, and one of the main reason that a lot of our families seek genetic testing. In order to counsel someone adequately about the recurrence risk of a genetic condition in their family, you have to first identify what the variant is. What is the actual cause of the genetic condition in that family unit? Once you're able to identify it, then there's the question of inheritance. And for dominant conditions, a parent that's affected has a 50% chance of passing it on to all of their offspring or each of their offspring. In a recessive condition, there's a 25% of passing it on if both parents are carriers. If only one parent is a carrier, then they're not likely to pass it. They're not expected to have an affected child, though half of their children will be carriers. And then in an X-linked condition, there's 50% of chance of passing it on with each pregnancy. But we see variability based on sex for who is more affected than others. So we typically see in X-linked conditions that males are more affected than females, but it remains that 50% chance of passing it on with each pregnancy. There are some other forms of inheritance that are not as common in the nephrology world, such as mitochondrial inheritance, but we didn't go through that today. It's not quite as applicable for this population. Genetic testing. So when a family wants to have answers prior to a pregnancy or during a pregnancy, there are some options there. Prior to conception, there are options to do in vitro fertilization with testing of the embryos. But this does require significant planning in advance and a high use of resources. It's also important to note that this is not something that can be done for every situation. In a child who has a de novo condition, so something that was new in them, and the parents want to know what the chances are of passing it on and want to have testing of embryos in the future, that is something that we're typically not able to do. So it can be very tricky, and it's important to counsel your families that they need to be speaking to a reproductive endocrinologist about their specific changes in order to move forward with something like IVF. There's also testing during a pregnancy, and this can include chorionic villus sampling, which is taking a small piece of the placenta, or amniocentesis, which is done by taking some of the amniotic fluid, and we can do genetic testing during the pregnancy. There's also the option of testing after birth. So in conclusion, I hope we've walked you through a little bit of how to read a genetic test report, the role of a genetic counselor, and really truly genetic counseling, and how you can integrate this into your practice. And then we've reviewed some really important genetic concepts in nephrology: mosaicism, some challenges with PKD1, genotype-phenotype correlation, and that's looking at the genetic change and seeing how it impacts the clinical aspects of somebody's care. Reduced penetrance and variable expressivity, so the differences within family members. We touched on digenic inheritance, and then gave you some tools to think about variants of uncertain significance and how to interpret those using functional studies such as metabolic labs, family studies, and ultimately collaboration with researchers. So I thank you for your attention, and I'll pass it back to Dr. Rastogi.
Dr. Russell, thank you so much. So that was an excellent, excellent overview of genetics, the primer for the modern nephrologist. So before we get into our next speaker, I just want to give a bit of a history of what we call Nephro-Genetics now at UCLA. So it was 2010 that we established the AD-PKD program at UCLA. At that time, PKD there's research being done, drugs, medications, but also more comprehensive care for a patient, because there are other organs that are also affected in AD-PKD. We can have aneurysms, we can have liver cysts, and other organs can get affected as well, including the cardiovascular system. So we started a comprehensive program for AD-PKD. A few years after that, we started a program for Fabry's disease, and then a few years Alport, and then kidney stones. And then finally, we put everything together. We said, we'll do a Nephro-Genetics program. And this Nephro-Genetics program would not have been possible without the help that we get from Dr. Bianca, Russell, and Marina. So we're very, very grateful for having them here at UCLA Health, but more importantly for our patients. So thank you very much, Dr. Russell. That was very informative, and thanks Marina, for your excellent presentation. One point that I do want to stress on what Dr. Russell went at the end is about family planning, and we should not miss that point. When a patient comes to, and I mentioned that in my opening remarks, why do the gene testing or broad panel testing for that matter? Number one is getting the right diagnosis, which is very, very important, because the management will depend upon that. And you'll be hearing from our guest speaker today about how the molecular diagnosis changed her life. Getting enrolled in clinical trials, a lot of trials going on. A lot of the medications that are coming out actually work by modulating gene expression, and a lot more will be coming out. But then also at-risk family members. So we should not be missing out on those that might be undiagnosed. And then finally, family planning. Every patient of mine who's in childbearing age gets automatically referred to Dr. Russell and Marina to get counseling on family planning for that matter.